This is a pretty long story, so I will try to cut it short. So let's go to the presentation. It is stuck here. Okay. So the focus of my presentation is osteoarthritis, which, as you know, is a complex degenerative and inflammatory disorder involving a bunch of etiological causative factors. Among the many, for example, cytokines are involved, such as interleukin-6 or matrix metalloproteinase. And osteoarthritis is a devastating disorder because, from the social sanitary point of view, it is very a lot of people suffer of osteoarthritis, especially the people above 50 years. And the other topic of my talk are extracellular vesicles, because among the etiological factors identified so far, also extracellular vesicles have been identified as promoting the onset and the degenerative process in osteoarthritis. And extracellular vesicles are involved in cells-to-cell communication in a plethora of events, but we will focus only on pro-inflammatory events promoted by extracellular vesicle in this pathological paradigm. And they contribute to inflammation in our setting, and also to cartilage degradation. And in osteoarthritis, another key factor is that the hyaluronic acid, whose mechanical characteristics are very important because they sharp the features, the biomechanical features of the joints. And hyaluronic acid, high molecular weight hyaluronic acid, serves as a cushioning and lubricating agent within the joints. Funziona? C'è un fischio. During osteoarthritis, hyaluronic acid is degraded into smaller fragments, and thus it loses its rheological properties and somehow contributes to the degenerative process. And to this end, the infiltrative treatment with high molecular weight hyaluronic acid has established as an important treatment to reduce the progression of the malady. So when hyaluronic acid is degraded, it loses these viscoelastic properties. And just take in mind this concept. Hyaluronic acid, okay, has been detailed by my colleagues before. And again, the importance of the molecular weight and the treatment of osteoarthritis. Infiltrated treatment with hyaluronic acid is based on the administration of linear high molecular weight hyaluronic acid, or also of cross-linked hyaluronic acid, because the cross-linked form has increased rheological properties and is more resistant to the degradation of hyaluronidases. We used a commercial blend of cross-linked and linear hyaluronic acid, which is somehow representative of the preparation that are on the market today. And which is the aim of the study? The aim of this study was simply to see whether hyaluronic acid is capable of interact with the bad extracellular vesicles released in the inflamed joint. And to this end, we used a pretty complex protocol, because first we had to decide which type of cells could be used to produce extracellular vesicles. And we focus on THP1 cells that can be activated in an inflammatory fashion, simply using, for example, interleukin-1, which is a cytokine involved in osteoarthritis. Then we stimulated THP1 cells to secrete extracellular vesicles, and we compared the extracellular vesicle from inflamed activated THP1 cells with the extracellular vesicle derived from neutral, non-activated THP1 cells. Then the target cells. We had to select the target cells, and we focused first on representative primary cultures because we isolated chondrocytes from patients undergoing surgery, osteoarthritis patients, Kellgren 4 grade of osteoarthritis, and we obtained primary cultures of chondrocytes which are representative of the malady. And then we exposed these chondrocytes to extracellular vesicles, both types, and we tried to see whether these extracellular vesicles were capable of inducing inflammatory responses in the target cells in the absence or presence of hyaluronic acids. This is the aim of the study. Again, no, I am going backward. And these are the results. First of all, we tried to see whether the two populations of extracellular vesicles, the activated ones which are identified as AV+, were somehow different from the neutral extracellular vesicles, which are called AV0. And this is a particle size distribution of the two populations of hyaluronic acid, of extracellular vesicles. And these are the neutral ones, and the inflamed, activated extracellular vesicles. It is not really indicative of differences between these two populations, but some difference, of course, is evident. And also what we tried to see was to identify the grade of interaction of extracellular vesicles with our target cells. And these small dots represent the internalized vesicles into the chondrocytes, the primary chondrocytes. So yes, we have two populations of extracellular vesicles, and they are capable to be internalized within the chondrocytes. And then we studied the effect of hyaluronic acid on target cells to see whether hyaluronic acid was toxic or not to our primary cultures, and we selected non-toxic concentrations of hyaluronic acid that was neither toxic nor capable of inducing any inflammatory response in the target cells. We measured the level of interleukin-6 in chondrocytes. Then the next step was to see whether extracellular vesicles were capable of inducing an inflammatory response in the target cells. And what we observed was that neutral extracellular vesicles obtained from non-activated THP1 cells were not capable of inducing any accumulation of interleukin-6, while the activated ones, activated extracellular vesicles, were capable of inducing a robust increase in interleukin-6, which is representative of an inflammatory activation of these cells. And interestingly, what we found was that hyaluronic acid, this mixture of cross-linked and linear hyaluronic acid, prevented the inflammatory induction caused by activated extracellular vesicle. The same effect was observed when we studied the accumulation of matrix metalloprotease 13, which was highly increased by treating chondrocytes with activated extracellular vesicles. And again, hyaluronic acid was capable of preventing this inflammatory response. So at this point, we had to characterize and see whether hyaluronic acid was capable of reacting and interacting with extracellular vesicle. And to this end, we had to develop a pretty complicated protocol based first on the preparation of biotinylated, cross-linked and linear hyaluronic acid. Then this biotinylated hyaluronic acid was incubated in the presence of extracellular vesicles. Then we prepared magnetic microbeads with streptavidin. Streptavidin reacts with biotin. Then we mixed 2 and 3, and we obtained probably a binding product between hyaluronic acid and microvesicle. And to see whether the binding would occur, we separated by centrifugation and magnetic separation the microbeads, and then in the pellet we tried to see whether we could also find extracellular vesicles. If extracellular vesicles are found in the pellet, it means that they had been captured by hyaluronic acid. And these are the results we obtained after this pretty complex procedure, but it was not easy to isolate hyaluronic acid and microvesicles. And this is the result we obtained. And this is the amount of microvesicles traces. We took TG101 as a hallmark of microvesicles. And these are the levels of extracellular vesicle found in the pellets. And as you can see, in the control we did not find any sign of extracellular vesicles, but increasing the level of extracellular vesicles against a fixed concentration of hyaluronic acid, we obtained a dose response related amount of extracellular vesicle in the pellet. So it means that somehow extracellular vesicle bind to hyaluronic acid. And then then we also tried to see whether the presence of hyaluronic acid was capable of preventing the uptake and the interaction of target cells with extracellular vesicle. And what you see here is that in the absence of hyaluronic acid, the extracellular vesicles are nicely internalized by chondrocytes, but in the presence of hyaluronic acid, there is almost complete prevention of the uptake of microvesicles. So there is no any more biological interaction between the target cells and the extracellular vesicles in the presence of hyaluronic acid. And I have no data because they are being calculated by Fabio in these very days, but we are trying to understand whether the point, the trigger of the binding between extracellular vesicles and hyaluronic acid is the CD44 receptor, which is known to react with hyaluronic acid, and it is also known to increase its expression, its membrane expression in THP1 cells when they are activated in an inflammatory fashion. And of course, when these cells secrete and release extracellular vesicle, they conserve in their membrane a high amount of CD44 receptor. So we think that the binding between hyaluronic acid and microvesicles take place at the level of CD44 receptor. But let's go straight to the conclusions and to the thoughts arising from this data. So the first one is that it is not a novelty, but we confirm that activated extracellular vesicles are capable of inducing and promoting quasi-inflammatory reaction in target cells. And so, yes, they probably contribute to the pathogenesis of osteoarthritis. And we also found that hyaluronic acid, both the cross-linked and the linear form, are capable of reducing and preventing the effects of extracellular vesicle on target cells. And to our best study, to our best knowledge, it is the first demonstration of this effect. And we have also an addendum to the pathogenesis of osteoarthritis. And this is a very simple view of our thought, of our hypothesis. On the leftmost of this slide, you can see what happens probably in an active inflammatory joint, where you have a release of a very high number of pro-inflammatory extracellular vesicle, and meanwhile, the hyaluronans in the synovial fluid are highly degraded into very small pieces, which are free to move within the joint space. But when you reconstitute the normal level and features of hyaluronic acid, injecting high molecular weight hyaluronic acid, then you have again a very solid network of the polymer, which is now capable of binding to extracellular vesicle, reducing their free movement within the synovial fluid and preventing the delivery of their cargoes to the target cells. And so this is somehow an addendum to the pathogenetic role of intact or degraded hyaluronic acid in terms of the progression of osteoarthritis. And the other remarks of our study is not this one, because this is an old version of my slides. I had no time to correct them, but it is very important to observe that today there is no rationale beside the formulation of hyaluronans for infiltrative treatment. The industry mainly produce high molecular weight blends of hyaluronic acid without any rationale. But now we think that we have somehow found one of the issues that may lead us to formulate new structure-activity relationships for hyaluronic acid, to understand which is the best blend to focus and to produce in order to obtain an optimal effect toward this type of interaction that in our hypothesis might be probably a topic one in the pathogenesis of osteoarthritis. So I beg your pardon because I did not speak about the topic of this meeting, because I spoke about osteoarthritis. So somehow I am off topic, but as you know, the bad joints lead to bad muscle function. So thank you very much.